So, hi everyone. Today I will not be translating exhibition art related things, but I will be talking about my PhD project. What I'm interested in is looking at the influence of cardiorespiratory fluctuations on one key component of bodily self-awareness, that is sense of agency. So today I'm going to present some very preliminary observation from the pilot phase of this project, which is currently ongoing in terms of data collection. So sense of agency refers to the experience of being in control of one's own actions and their consequences in the environment. And traditionally, at least at an implicit level, sense of agency has been operationalized via this intentional binding effect. And so we see that the intentional binding effect refers to the perceived temporal compression of the interval between a voluntary action and its subsequent sensory outcome, in this case, a tone. So what happens is that the action is perceived to be later in time than its actual occurrence, so closer to the outcome, and the outcome is perceived to be earlier in time, so closer to the action that generated it. And this is in comparison with baseline conditions in which the action and the outcome occur in isolation from one another. So the idea that interoception might play a role in sense of agency arises from certain predictive models that posit the role of interoceptive signals in the generation, the control and self-attribution of motor actions. And so on one hand, we have indeed evidence of the first part, so that interoceptive signals might play a role in the generation of actions, in the sense that we see that there is an increased tendency to initiate voluntary movements in specific phases of the cardiac and the respiratory cycle. Mainly we see this effect in early cardiac phases, so during systole, and in late phases of the respiratory cycle. However, when it comes to the role of interoceptive signals in sense of agency, the evidence is less clear and a bit mixed. So for this reason, in this first project, the idea is to investigate whether these preferential time locking that we see of voluntary movements in specific phases of the cardiac and the respiratory cycle might in turn influence sense of agency, so the resulting experience of being in control for those same actions, as indexed by the magnitude of the intentional binding effect. And also seeing whether there is an effect of whether the action and the outcome fall in the same or different cardiac and respiratory phases, with the idea that different phases might be more or less beneficial for action or for perception. And lastly, the combined phase locking of these two rhythms, what effect does it have on sense of agency? So just to give you a brief overview of the task without going into details, but what participants do is that they are asked to, in baseline conditions, they are asked to either press a key at a time of their own choosing, or they wait for a tone to be played, and then they report a time when these events occurred in isolation. Otherwise, in what I call operant conditions, the action always triggers a tone, 250 milliseconds afterwards, but in one condition, participants only focus on the time when the action occurs, and instead, in another condition, they focus on the time of the outcome, so the time of the tone. And this while we are currently recording ECG and respiratory measures. So for this pilot sample, the one good thing is that indeed we see intentional binding effects. So at least at the behavioral level, everything is good. We see that indeed the action is perceived to be later in time and the tone is perceived to be earlier in time. So both components of action binding and tone binding are there. You can see this also in the distribution of their judgment errors. So by how much these actions or outcomes are perceived with respect to their actual time in which they occur. And then I just wanted to, without also going into much details, to mention that so by now what I've done is to develop this pipeline for the pre-processing and analysis of the ECG in relation to behavior. But this has been possible thanks to the amazing people that have been working with me in the brain-body analysis special interest group, which is a collaborative initiative that myself and my supervisor, Michael Gebler, have initiated with both people from the lab, from the department, and extended also to other groups, in which we try to develop these pipelines in open source languages, so mainly in Python, using also existing packages, but also integrating our own functions. And so, yeah, just to give you an idea of the analysis that I would like to then do moving forward with the actual sample from data collection. Circular analysis, so in this case, trying to look at the action onset across the cardiac cycle. So in this case, we see examples from individual data. We see that people, their mean, the circular mean and mean resulting length vary across the cardiac cycle when they initiate the actions. And then we derive basically these mean values to do between subject second level analysis. And then binary analysis. So in this case, we use an individualized approach to segment the cardiac cycle into systole and diastole using parameters that are taken from the QRS complex, from the T-wave end, etc. And we then use that to bin action onset and tone onset onto the cardiac cycle. And so in this case, then looking at how the action binding and tone binding components are modulated as a result of whether the action was, for example, initiated during systole or diastole. I'm not talking about the results because this is very like from the pilot, so we cannot tell anything. It's just also to give you an idea of the analysis plan that I intend to do. So the very take-home message from now is that we see that the ECG behavior analysis pipeline and the behavioral results per se are in line with prior literature, and this is work in progress. So we are currently ongoing data collection and developing the pipelines for the respiratory data and the bimodal cardiorespiratory analysis. If you're interested to know more, the study is pre-registered, so you can look at the OSF pre-registration, but you can also feel free to contact me either via email or on social media. And thanks obviously to my supervisors and to the Mind Body Emotion group for supporting me in my PhD journey.
Thank you very much for the talk. Are there questions?
Thank you, super cool. I was just wondering, maybe I missed it, maybe you said it. What would be your prediction for the contraction? So would you kind of expect it to be at systole?
Yes, so that's a good question. So the prediction is on one hand to replicate what has been shown in previous studies, so that there is an increased tendency to initiate action during systole and during the late phases of expiration. And then on the other hand, what I expect on sense of agency is that since systole and the expiration is less clear, but systole is more related to action initiation, then sense of agency resulting from actions initiated at systole over diastole results in greater intentional binding, so a greater temporal compression.
Yes, I guess it's really interesting for me because time perception and kind of to see whether you actually see the contraction of time, and obviously then it would be stronger in the sense of agency. Yeah, exactly. Thank you.
Yeah, that's quite cool. I mean, I read your study and I was thinking about that quite a lot. I also think that one future development could be to look at different temporal relations, in the sense that we know that sense of agency decreases when the interval increases between the action and the outcome. And so looking at whether also different fluctuations, so whether the cardiac and the respiratory rhythm, there are in different scales, modulate differently these relation in terms of sense of agency for shorter or longer intervals.